In a previous presentation, we learned about the cell cycle and how cells grow in G1 phase, synthesize DNA in S phase, grow again in G2 phase, and finally divide during the M phase. How does a cell know when to do each of these phases? How does it know when to enter mitosis? A series of experiments in the 1970s sought to determine whether the cell cycle is controlled by chemical signals. In these experiments, and others like it, cytoplasm from one cell was injected into the cytoplasm of another cell. In the experiments on the left, the cytoplasm was taken from a cell undergoing mitosis and injected into a cell in interphase. For the cell on the right, the same experiment was done, except cytoplasm was taken from another interphase cell. Thus, all the same experimental procedures were carried out in both cells, except for the timing of the cell from which the cytoplasm was taken. The prediction was that if there were molecules that were controlling the entry of a cell into M phase, then injecting M phase cytoplasm, like the cell on the left, would cause the cell to go into mitosis. If the cell cycle was not controlled by regulatory molecules, or those molecules didn't exist in the cytoplasm, then neither cell would enter mitosis. When this experiment was carried out, the cell on the left immediately entered mitosis, and the cell on the right did not. This showed that there were regulatory molecules in the cytoplasm of the M phase cell that initiated mitosis. Similar experiments were done that showed that cytoplasmic molecules also control the cell's entry into S phase and the synthesis of DNA. So let's look at how these molecules regulate mitosis and then look at what exactly they are. You can think of the cell cycle as containing a number of checkpoints. These are points of the cell cycle where certain conditions must be met in order for the cell cycle to progress past that point. These checkpoints are controlled by external and internal conditions, which we'll look at in a few minutes. The cell's entry into M phase is largely controlled by a protein complex called MPF, or maturation promoting factor. MPF is itself made up of two components, cyclin and a cyclin-dependent kinase. If you remember back to our discussion of cellular respiration, a kinase is an enzyme that adds a phosphate group to another molecule. This phosphorylation can activate or deactivate a second molecule or a target protein. The cyclin-dependent kinase, or CDK, has a constant concentration during the entire cell cycle. On its own, it's inactive. It needs another molecule in order to become an active kinase. That other molecule is cyclin, which, as its name suggests, cycles in concentration according to the cell cycle. During the G2 phase, cyclin production in the cell ramps up considerably. Once the CDK cyclin complex, or MPF, reaches a threshold, it becomes very active, and the cell successfully passes the G2 checkpoint and enters M phase. Toward the end of M phase, cyclin is degraded and the CDK becomes inactive without it. What's shown here is the CDK cyclin activity of one pair of CDK cyclins. In fact, there are several different cyclins which control passage of different checkpoints along the cell cycle. Let's take a look at some of those other checkpoints. One of the most important checkpoints is called the G1 checkpoint. As I mentioned earlier, many cells exit the cell cycle into a phase called G0. While in G0, these cells are just doing the normal thing that these cells do. In other words, they are not replicating. You can see from this picture a number of signals are required in order for the cell to pass the G1 checkpoint. First, the cell must be large. Remember that one of the purposes of the G1 phase is for the cell to grow. If it hasn't grown large enough, then it won't be able to pass this checkpoint. The cell also needs to have available nutrients. The next phase of the cell cycle is DNA synthesis, and this phase is energetically demanding and requires enough raw materials to make new strands of DNA. Therefore, the availability of nutrients is important. The cell may also be required to receive chemical signals before it can divide. One such chemical signal is growth factors, which are proteins released by some cells as a signal to others that those other cells should divide. Social signals are also used to pass the G1 checkpoint. Usually, cells don't like to pile up on one another. That is, they exhibit density-dependent inhibition. Cells are able to sense the number of neighbors they have and usually will not divide if they are already crowded. Cells also like to be firmly anchored to the extracellular matrix, and this anchorage dependence reduces the chance that they will divide unless they're attached. Finally, damage to DNA will also prevent the cell from passing the G1 checkpoint. P53 is a tumor suppressor gene, and the protein that is produced by it checks for DNA damage. If extensive damage is detected, the G1 checkpoint won't be passed. If the cell passes the G1 checkpoint, it goes on to synthesize new DNA in S phase and then grow again in G2. Before it finally commits to dividing, it needs to pass the G2 checkpoint. The G2 checkpoint is the last time the cell has control before entering into mitosis. In order to pass into mitosis, the cell checks that the DNA is not damaged and that all of it is replicated. All this is controlled by CDK cyclin activity. When everything is ready to go, inhibitors of CDK cyclin, which we haven't talked about yet, are removed, and this greatly increases the CDK cyclin activity, allowing entry into mitosis. In order to pass from metaphase into anaphase, the cell checks that all kinetochores are properly attached to the microtubules. Without proper attachment to microtubules, it's possible that some sister chromatids will be dragged together into one cell, resulting in a daughter cell that has 2 copies of a chromosome, and another daughter cell that doesn't have any copies of that chromosome. Therefore, checking that the kinetochores are attached properly is important to maintain equal distribution of chromosomes in the daughter cells. Finally, to pass from anaphase into telophase, the cyclin needs to be fully degraded, and this happens after chromosomes are completely separated. If they don't separate completely, the cell won't complete anaphase until that happens. So, the checkpoints we've been looking at are essentially a way that cells put a brake on cell division. Notice that several of these checkpoints involve checks that the DNA is undamaged and properly replicated. In order to check this, however, there need to be genes that produce gametes to do the checking. That produce proteins to do the checking. Sometimes, genes are not copied completely perfectly, resulting in a mutation. If this mutation happens in a gene that controls the checkpoints, then the checkpoints may fail. If checkpoints fail, then the cell will progress through the cell cycle much more quickly and without doing all of the normal checks that usually guarantee that only healthy cells reproduce. For example, P53 is a gene that produces a protein that's also called P53. The protein P53 is responsible for checking that DNA is not damaged. Normally, if damaged DNA is detected, P53 activates other genes that causes the cell to self-destruct, a process called apoptosis. However, if there's a mutation in P53 that causes it to not function properly, then damaged DNA will not bring about apoptosis, and the cell cycle will continue even in the presence of damaged DNA. So, when control of the cell cycle breaks down, the cell cycle progresses more rapidly and without the checks that normal cells show. As the cells reproduce more and more, additional mutations are introduced, which further reduce control of the cell cycle. This lack of control of the cell cycle is a common disease
that we know as cancer. When a cell loses control of the cell cycle, it starts to reproduce more quickly. And it happens in inappropriate places and at inappropriate times. It also passes on its defective genetics to its daughter cells, which then reproduce at a slightly faster rate than the neighboring cells. Even a slight increase in the rate of reproduction of these cells can, over time, lead to the development of a tumor. Often, tumors are benign and do not cause health problems for the individual. However, additional mutations may cause cells of the tumor to be able to survive and grow in other parts of the body. When this happens, the tumor is said to be malignant, and it metastasizes to other parts of the body, often through blood vessels or the lymphatic system. Treatment for cancer usually involves surgery to remove cancerous cells, alongside either chemotherapy or radiation therapy. Chemotherapy involves toxic chemicals that interfere with the cell cycle. Because cancer cells are essentially ignoring the checks on the cell cycle, chemotherapy provides a strong brake on cellular reproduction. The problem, however, is that chemotherapy also affects normal cells, and this is why it's so toxic. Usually, the goal of a doctor treating a cancer patient is to kill all of the cancerous cells, but as few of the normal cells as possible. And this is a difficult balance to strike. In recent years, other therapies have become available, including therapies that involve strengthening the immune system and training the immune system to detect and eliminate cancerous cells.